To put our central metabolism in context, we will generally be talking about the path of energy all the way from a food that contains a major biomolecule to its conversion to acetyl-CoA. Once we get to acetyl-CoA, the catabolic pathways to energy are the same for all biomolecules. We go through the citric acid cycle, which is also called the Krebs cycle after the Nobel laureate who did much of the initial studies on the pathway. It's also known as the tricarboxylic acid cycle, or TCA cycle, because why not? I'll use citric acid cycle, and so does your book. The citric acid cycle is cyclic. It begins and ends with the same molecule, so it's actually what happens to the acetyl-CoA and the byproducts of the cycle that are important. Those byproducts take energy harvested in the citric acid cycle into the electron transport chain, so both pathways are connected. Again, the very big picture is that we will, for any biomolecules, break dietary forms into simpler forms that can enter our bloodstream, like fatty acids, glucose, and amino acids. Each of those pieces can be used in a variety of ways, but when they're used for energy, they become acetyl-CoA and are processed through the citric acid cycle and the electron transport chain. And those are the main focuses of this chapter. We're going to go into specifics about the citric acid cycle in a little bit, but the most important thing to know here is the overall reaction. For my purposes, you can ignore the Pi, which is inorganic phosphate, the water, and the H+. Note that most of the reactants are paired. Acetyl-CoA comes in, 2 carbon dioxides and an unloaded CoA go out. 3 NAD+ go in, 3 NADH come out, FAD goes in, FADH2 goes out, GDP goes in, GTP comes out. In this cycle, the acetyl carbons are oxidized to carbon dioxide and all the redox cofactors are reduced. The carbons coming in bring the energy, and the energy leaves ready to be used as GTP or as NADH and FADH2 that can be processed for energy in the electron transport chain. The citric acid cycle is an example of metabolic regulation through allosteric enzymes as well. Citrate synthase, which is the first enzyme we'll talk about, is regulated by feedback with ATP. Since ATP is the end goal for the citric acid cycle and the electron transport chain, having lots of ATP slows down the cycle. The third enzyme, isocitrate dehydrogenase, has 2 allosteric regulators, one that's negative and one that's positive. The 2 regulators are NADH and ADP. Which do you think is a positive regulator, or that is, which of these do you think will make the citric acid cycle go faster? The citric acid cycle is a cyclic pathway, in case that wasn't obvious from the cycle part of the name. The citric acid cycle begins with acetyl from acetyl-CoA being joined to oxaloacetate, and the remainder of that cycle is oxidizing those carbons off and returning the structure to oxaloacetate. Many of the most important steps are oxidations. This is where we generate the cofactors NADH and FADH2 that can be used for energy elsewhere. The first step of the citric acid cycle is considered a condensation. We join 2 pieces together to get the acetyl-CoA carbons into the cycle. The second step of the citric acid cycle doesn't give us any energy, but it does posture us for getting energy at a later step. Moving the alcohol allows us to harvest more energy later, since we can oxidize that alcohol at its new location. It moved from being a tertiary alcohol to being a secondary alcohol. Step 3 is an oxidation and a decarboxylation. The oxidation is really important because it uses NAD+, meaning we need to have a supply of NAD+, and because it produces NADH, which can be used for energy if we have oxygen. Once the alcohol is oxidized to a ketone, reactivity that we don't talk about in this course allows for the carbon dioxide to easily be removed. This is important because it allows the neighboring carbon, which was tertiary, to become secondary and more easily oxidized. The fourth reaction is another oxidation. It's harder to see because it is actually a ketone that gets oxidized, which we rarely see. When the ketone is oxidized, it releases the carbon dioxide and is trapped as a thioester, which is a derivative of a carboxylic acid. Notice that the oxidation uses another NAD+ and produces another NADH. We're now at a total of 2 NADHs produced that will account for some of the energy we get from the citric acid cycle. The succinyl-CoA is trapped in a higher energy state from the oxidation in the previous step. Step 5 is the only step where an energy-carrying product, in this case GTP, is produced without oxidation. And that's only because the energy came from a leftover in the oxidation in the previous step. All the energy in the citric acid cycle comes from oxidation and is stored in reduced cofactors. After this step, we're at a total of 2 NADHs that have been produced in the cycle and 1 GTP. The next step is the first of a sequence of reactions that are really common in biology, an oxidation from an alkane to an alkene, that's then followed by hydration of that alkene to an alcohol, and then followed by another oxidation. Again, this first step of that common sequence converts an alkane to an alkene. FAD is the preferred redox cofactor for oxidizing those nonpolar CC bonds. So going from alkane to alkene is almost always FAD. So we use FAD and we produce FADH2 in this reaction. Now we're at a total of 2 NADHs, 1 FADH2, and 1 GTP produced in this sequence. Hydrations are not oxidations. The alkene is just as oxidized as the alcohol, but it's hard to oxidize an alkene any further, but it's easy to oxidize an alcohol. So the hydration step at step 7 allows us to posture ourselves for additional oxidation. Our final reaction is another oxidation, giving us a final NADH and restoring oxaloacetate, which we can now react with another acetyl-CoA. Again, don't memorize the individual steps of the citric acid cycle, but do know what the overall reaction is and what types of steps are giving us energy in the form of NADH and FADH2. Hint, it's oxidation. I just want to really briefly revisit this slide now that we've gone through the citric acid cycle. Again, our general goal when converting food into energy is to break it down and absorb the simple forms of each type of biomolecule into the bloodstream. Each type of simple biomolecule may be broken down into acetyl-CoA, which then gets processed in the citric acid cycle. This cycle is connected to our next process, the electron transport chain, by the redox cofactors NADH and FADH2 that are produced in the citric acid cycle. Again, 3 NADH and 1 FADH2 are produced in the citric acid cycle, and they will be used in the electron transport chain.